वेलकम बैक माय डियर स्टूडेंट्स। हम आज बात करने जा रहे हैं सीड की। तो पहले हम देखेंगे सीड क्या है, हाउ वी कैन डिफाइन सीड। उसके बाद हम देखेंगे सीड के कंपोनेंट्स क्या होते हैं, इसमें क्या क्या होता है। एंड देन फाइनली वी विल डिस्कस द टाइप्स ऑफ सीड्स ऑन द बेसिस ऑफ प्रेजेंस और एब्सेंस ऑफ एंडोस्पर्म। टेक्निकली हम सीड को डिफाइन कर सकते हैं। It is defined as a fertilized and metamorphosed ovule containing an embryo enclosed inside it. And this seed, it is having resistant protective coats. It contains seed coats which are resistant. A seed is defined as a fertilized and metamorphosed ovule containing an embryo enclosed in it. We can also describe seed as it is the initial dormant stage of the diploid sporophytic phase of the diploid sporophytic generation, you can say, in the life cycle of spermatophytes or phanerogames. जो सीड होता है ये इनिशियल स्टेज होता है, इनिशियल बट डॉर्मेंट स्टेज. इट इज द इनिशियल बट डॉर्मेंट स्टेज ऑफ द डिप्लॉयड स्पोरोफिटिक जनरेशन. जो स्पोरोफिटिक जनरेशन होती है स्पर्मेटोफाइट्स में या फिनेरोगेम्स में, हम कह सकते हैं इट इज रिप्रेजेंटेड बाय द सीड. द इनिशियल डॉर्मेंट स्टेज in the life cycle of the spermatophytes, it is represented by the seed. Now you know the seed, it is the final product of sexual reproduction in angiosperms. Okay, it is a final product of sexual reproduction. जब sexual reproduction हो जाती है, fertilization हो जाती है, उसी के बाद seed बनता है. So a seed develops from fertilized ovule which is lodged inside the fruits. फ्रूट्स के अंदर ओव्यूल्स होते हैं. व्हेन दे गेट फर्टिलाइज्ड दे बिकम सीड्स. A seed consists of cotyledons, seed coats and an embryonal axis. यहाँ पे आप देख सकते हैं, this is the seed. Here you can see these are the two cotyledons. This is the embryonal axis consisting of pillimule or future shoot and radicle or future root. Now the types of seeds based on the presence or absence of endosperm. You already know there are two types of seeds on the basis of the presence or absence of endosperm. The first category is non-albuminous or non-endospermic. Those seeds in which the endosperm is utilized during the embryo development. That means all the endosperm which is present in the seed, it is utilized by the embryo during its development. Such seeds are known as non-albuminous or non-endospermic seeds. And the best example that you can remember here is peanut and groundnut. Okay, so non-endospermic seeds or non-albuminous seeds are those seeds in which all the endosperm is utilized, is consumed by the developing embryo. Okay, and there is no endosperm left. The second one is albuminous or endospermic seeds. Albuminous or endospermic seeds, those seeds in which some part of the endosperm is still present when the seed becomes mature. Such seeds are known as albuminous or endospermic seeds. And the examples here are wheat, maize, barley, castor and sunflower. Here you can see this is the grain of maize. Okay, a maize grain. This portion here, pink portion, it consists of the embryo. It represents the embryo. And this yellow portion, this broad yellow portion, it is the endosperm. Okay, now an important point here you can remember, occasionally the nucleus, जो diploid tissue होता है seed का, ovule का, okay, occasionally it has been found that nucleus persists. जो न्यूक्लियस होता है, जो डिप्लॉयड टिश्यू होता है सीड का, ओके, it has been found that in some cases the nucleus persists and it has a nutritive role. This persistent nucleus, it is known as perisperm. मैं आपको दिखा दूं यहाँ पे. For example, this is a seed, okay. Inside it you have the embryo sac, then this is the PN, it will form the endosperm, it will form endosperm. Here you will have the embryo here, okay. And when the endosperm it expands, this diploid tissue it gets consumed, ये खत्म हो जाता है. But in some cases it has been found that the diploid nucleus, the diploid nucleus tissue of the ovule, it persists till maturity of the seed, and this tissue is known as perisperm, okay. This is known as perisperm and it has nutritive role. So perisperm is the diploid persistent nucleus present in the seeds of some plants, example black pepper and beet. You can find perisperm in the seeds of black pepper and beetroot. Now the seed coats. The seed coats, they are the outermost protective coverings. They develop from the integuments. The outer seed coat, it forms testa. It is known as testa. And the inner one is known as tegmen. So you can say that the outer integument forms testa, and the inner integument of the ovule it forms the tegmen. Once again I will repeat. The seed coats, they develop from integuments of the ovule. The outer integument forms testa and the inner one forms tegmen. Now the micropile. What is micropile? Micropile is a small pore in the seed coat, and this micropile helps us in the movement of water and oxygen during germination process. जब हम seeds को sow करते हैं, okay, when you allow seeds to grow in the soil, you sow seeds, they possess a small opening here, okay. They possess a small opening or pore. This pore is the micropile. The function of micropile is it allows the movement of water and oxygen during the germination of seed in the soil. Now seed maturation. How seed becomes mature? Seed becomes mature by reduction in water content. Okay, so at maturity you will find the seeds contain 10 to 15% of water of their dry weight. Okay, it reduces to 10 to 15% of dry weight of the seeds. So simply stated, you can say when seeds become mature, there occurs reduction in its water content. The second one is the metabolic activity slows down. That means the embryo it becomes dormant or inactive. When seed maturation occurs, the embryo it becomes inactive, or you can say its metabolism becomes slow. It gets metabolically inactive, okay, for a period for some time period. Then finally, when the seed has passed all the stages, okay, it has reduced its water content, the embryo has become dormant or inactive, then after some time interval the seeds are capable of germinating. When the favorable environmental condition is then returned, that means adequate moisture is there in the environment, temperature is optimum, and there are enough oxygen levels, okay, then the seed can germinate. So you can remember, when seed maturation occurs, its water content gets reduced, its metabolic activity slows down, that means embryo becomes dormant or inactive for some time period. Then finally, when the favorable environmental condition is returned, that means there is adequate moisture, there is optimum temperature, and oxygen is abundant, the seed can germinate and give rise to new plant. Here you can see we will have uh, we will an overview of the structure of seed. This is the cotyledon. Okay, it is this small structure, it is the micropile, and this structure here, this is the embryo. Okay, this embryo consists of the embryonal axis having pilumule which forms the future shoot, then there is hypocotyle, and at this end we have the uh, radicle which forms the future root of the plant, and the whole structure, it is covered by the integuments. Two integuments, outer integument is known as testa and the inner integument is known as tegumen. Okay, so this was all about the structure of seed and types of seeds. See you soon with a new lesson. Till then, take care.